Ladies and gentlemen, good evening and a really warm welcome on a cold evening to Anglia Ruskin University. My name is Roger Watkins, I'm Vice-Chancellor. I'm just delighted to see such a great turnout this evening. You're here for an inaugural lecture, not just an inaugural lecture, but Havi's inaugural lecture on an amazing topic. This is a really happy event for us, it's a celebratory event for us. Inaugural lectures are always really exciting celebratory events in universities where we recognise the outstanding achievements of colleagues recently joined us or colleagues whose career has flourished at ARU and they have gone on to secure professorship in recognition of their outstanding contribution. So I'm very much looking forward to learning more. I'm hoping we get to eat some of the illustrated sweets. My pleasure, first off, though, is to invite Professor Laurie Butler, Pro-Vice-Chancellor and Dean of the Faculty of Science and Engineering, to properly introduce this evening's speaker. So please join me in welcoming Laurie.
Thank you very much, Vice-Chancellor, and a welcome to you all. So it gives me absolute pleasure to introduce tonight's speaker for this, the first of our FSc inaugural lectures for this academic year, and I'm really pleased that it is one of many inaugural professorial lectures from colleagues in FSc over the course of the next few months. So Professor Havovi Chichger is a Professor of Biomedical Science and a Deputy Head within the School of Life Sciences. Havovi started her academic career with a PhD at UCL, focusing on glucose transport in metabolic disease and diabetes. She then undertook her postdoctoral fellowship at Brown University in the US, where she expanded her research scope to investigate the physiological mechanisms linked to cardiovascular disease. In 2015, Havovi joined ARU as a senior lecturer, where she focused on developing our understanding of the cellular mechanisms regulating blood vessel function. Through her research, Havovi has identified a new family of proteins, taste receptors, which play a key role in cardiovascular disease, and she's currently investigating how to unlock their therapeutic potential with a clinical trial in the US. Havovi has published more than 40 papers in high-quality journals such as the European Respiratory Journal and Pulmonary Circulation. Her research has been cited by the WHO, and the UK government published guidance on artificial sweetener use based on her work. Havovi has won various awards for her research, including the Judah Folkman Services to the field of vascular biology in 2024 and the Experimental Physiology Career Research Award in 2022. She's widely recognised as an exceptional mentor, offering insightful guidance and unwavering support to both colleagues and students. She's successfully supervised 15 PhD students to completion and is currently supervising 9 further PhD students, both internally and externally to ARU. Now, within ARU, alongside Havovi's very busy research portfolio, Havovi has also led a range of modules in the biomedical science discipline. She's been course director for the MSc Applied Bioscience degree and sat on a variety of committees, including the Faculty Athena Swan team and the university's Women's Network Steering Group. She's about to complete her 4-year tenure as co-deputy head of school with Claudia Vascia, where she not only helped drive forward the School of Life Sciences research strategy, but also pioneered the job share model of leadership here at ARU. Externally, Havovi contributes as editor to 6 journals. I did double check, 6 journals. Wow. Including Experimental Physiology and the American Journal of Lung, Cellular and Molecular Biology, and is also a member of various expert panels, including the WHO Nutritional Advisory Committee and grant review boards for the MRC and also for the NIH as well. Havovi also contributes to public engagement through public workshops at the Cambridge Festival every year, and her research has been covered by a range of newspapers, including The Independent, USA Today, The Telegraph, and Naked Scientists podcast. Havovi's personal career highlight, after all of that I've just told you, was being interviewed by Gloria Honeyford for Food, Truth or Scare, a BBC television programme. You can ask Havovi about that later on. And in her spare time, Havovi is a keen baker and a recent, although she says slightly begrudging, convert to home renovation and DIY projects. So in her inaugural lecture tonight, Havovi will tell us more about her journey through academia, reveal some of the lessons she's learned, and give us a taste of how her research is uncovering new potential therapeutic approaches to cardiovascular disease. So, with a talk entitled Sweet Signals, Bitter Truths, Taste Sensing and Cardiovascular Disease, it's my absolute pleasure to introduce Professor Havovi Chichger.
Thank you very much. I can't tell you how honoured I am to be standing here talking to you about my research. And as you can tell from the image, as Roderick mentioned, a lot of it is around sweet taste. So I felt it imperative to have an image for you. In fact, Laura encouraged us to have props. So I really wanted to bring sweets along for you all to have a little kind of taste throughout. However, I also researched diabetes, so it felt inappropriate to be giving out sweets today. But I'm sure there'll be lovely refreshments after the session that you can enjoy. So instead, I'm going to tell you about how I went from this to the awesome professor you see in front of you. And I have to take a quote from a Nigerian community about it takes a village to raise a child. Well, it definitely took a village to raise this professor. And I hope today to be able to thank many of the village that raised me as I talk through the sort of slides and talk through my journey. And it really started with this. So those of you who are a child of the 90s or 80s might get PTSD seeing this. For those of you who don't remember or don't know, this is a national record of achievement. So age 11, Havovi had to complete this, and it was all about what she wanted to be. So everyone wanted to be footballers or maybe astronauts. I put in this that I wanted to be a professor in biomedical science. So me standing in front of you today is either tenacious, hard-working, or just a lack of imagination and creativity. Either way, here I am, and I'll take the win. So I was always destined to be a scientist. My mother is a biochemist by training, my dad an engineer, and whilst my sister Navaz followed in dad's footsteps and went into construction, I decided to follow mum's footsteps and go into biochemistry. And again, lack of creativity, lack of imagination, but I did love biochemistry, so again, we'll take the win. Sadly, Dad's not here today. He died a couple of years ago, but since I had my PhD, since I got my PhD, he has treated me like the most esteemed professor in the field. So I've no doubt he would be absolutely delighted to be here today to celebrate. So I went off to King's College. I did my degree in biochemistry. I really enjoyed it, really got into the kind of nitty-gritty of it. And in my second year, I got the opportunity to do an internship at MSD, which I think is now changed or been bought out, but Merck Sharp Dome, it was a pharmaceutical company. So I had a great summer working in a pharmaceutical company, and I realized it wasn't for me. Perhaps a bit reductionist, perhaps a little bit naive at the time, but I knew I wanted to go back into academic research. And so in my third year, in my dissertation project, I studied this protein, P53. So this was a protein all about cancer. There was a big buzz at the time around it because it was mutated in a lot of different types of cancer. And my project was all about looking at this. And I loved getting in the lab, getting to ask a research question, design an experiment, answer it. Usually the answer wasn't what you wanted to see, but hey, that's science. And so I came out of my degree with some very good friends, some great experiences, and a love of research. So at that time, I was really lucky. It was a time where fully funded PhDs were left, right, and centre. So I went off to University of Edinburgh on a BBSRC funded PhD and studied this protein that I've become so interested in, P53. And so what we were looking at was how the structure of P53 changes, how it adapts and moves when it binds to different types of molecules, and how that makes it more active or inactive, and how that might link to cancer. But a lot of that was working with test tubes. So never got to see any models, never got to do any work outside these sort of small protein test tubes. And whilst that is a fantastic area of science, I realized it wasn't quite for me. And so we got a paper out of it, which was great, but I sort of realized that the fit wasn't right. I wasn't really happy there. And it's always a bit of a difficult thing to try and talk about this, because I think as scientists and as career academics, we love to talk about our fantastic journeys. But I think it's important to say sometimes it's a bit windy, things don't always work out. So I stepped back from that PhD and knew that I wanted to stay in research, but maybe this wasn't the right field for me. I moved back down to London, and I was lucky to join a fantastic lab at UCL. So I did my PhD number 2. It was funded by Diabetes UK. And I was supervised by Ted Debnam, Robert Unwin, and Joe Marks. And this was where I belonged. To say that it fit, to say that physiology felt like the right fit to me, is an understatement. We were looking at diabetes, and we were looking at kidney disease, specifically in diabetes, and trying to understand how the kidney can handle glucose and how it goes wrong in patients, and trying to avoid sort of the end-stage renal disease and dialysis that comes to patients with sort of uncontrolled diabetes. And so we were looking at the kidney. So I'm going to take you back to your basic biology. This is a nephron. It is in your kidney. And at the start, my pointer's not going to work. So early on in the kidney is where we absorb all our glucose. And so this is the area we were focused on. We were interested in looking at how that absorbs. So if we think about a normal person, here we have on the top side, we have how our blood filters into the kidney, and on the bottom side, we have what's absorbed. So in a normal individual, you have your glucose coming through, you have your glucose transporters, and they will take all of the glucose. This is how we make sure that in a normal individual, we don't have glucose in the urine. But in patients with diabetes, and specifically uncontrolled diabetes, they have a lot of glucose present, and it can't be handled. So we end up with a scenario where these patients have glucose in their urine. And around the time that I was working on this, there was some really nice work showing that if you block one of the glucose transporters, it would help patients. It would mean that not so much glucose was reabsorbed into their blood, more goes in the urine, but that was good. It brought their glucose down. But we were interested in GLUT2, in the other transporter. And so through my PhD, we looked at a variety of different physiological models of diabetes. We looked at a way to induce diabetes by killing beta cells. We also looked at a high-fat and Mediterranean, Western diet to try and understand the kidney. So we took the kidney out. I apologise right now. I'm going to put you off kidney beans for a little while with this image. I was put off for years after. I can only now start to eat them again. But we took the kidneys out, and we took out specifically this region of the nephron that we were interested in, where glucose transport happened. And we found various different ways that these transporters were managed. So if we think about our patient with diabetes, we found this receptor, or these two receptors, taste receptors, that were, when activated, could promote the amount of glucose that was absorbed. So this was sort of a nice new novel finding. It was particularly interesting because these are the taste receptors that we normally find in our mouth. So these are the taste receptors that sense sweet things and tell us something tastes good, so we want more of it. These are the taste receptors that tell our brain that there's food coming in, that there's carbohydrates coming in, that we need to increase our digestion. And it was around this time that sort of some researchers were finding these in different locations, but we were the first to find it in the kidney. So I was really enjoying this. I was loving being a physiologist, but as always happens, I got right into the science, and it was time for my PhD to end, and I had to write up my dissertation. And so I was looking for postdoc opportunities, and I'd seen some interesting work on the kidney vasculature, so the blood vessels surrounding the kidney, and thought that was interesting. Met with a couple of PIs, and I ended up in America. Now, I was very fortunate to be in America in the Obama years. I packed my suitcase, or multiple suitcases, as the case might be, and started a funded postdoc funded by the NIH at Brown University. And specifically, we were based in a Veterans Affairs, Veterans of America Medical Center. So that's where the research was based. It was very clinical focused. And I was supported and supervised, mentored by three incredible researchers, Professor Beth Harrington, Professor Sharon Rounds, and Professor Gaurav Chowdhury. And although my aim had been to look at kidney blood vessels, I very quickly got drawn into lung blood vessels. And this is where I sort of really got excited by the research that was going on. And the nice thing was, because we had that sort of clinical side right there, that we had the kind of constant patient-clinician interaction. And so I want to tell you a bit about this disease that I got interested in here, because it sort of formed the basis of where my career has gone. So it's called acute respiratory distress syndrome, and this is what happens to patients. This is what their lungs look like. So in this slide here, you would expect a nice, round, open alveoli to be clear, to be white, so that it allows gas exchange to occur. But in patients with ARDS, it's filled with water. So it's that water on the lungs, or pulmonary edema. And when we look at a scan, or a CT scan in this case, we can see these sort of white patchy areas. So where we'd like to see a nice black image here for where the lungs should be, they're sort of patchy. And what it means is that patients can't breathe properly. They become very breathless. They have very little oxygen in their blood. It can happen from infection, but in the case that we were looking at, it happens a lot in trauma. So in veterans, typically who've had a lot of trauma, they can be much more susceptible. It also can happen with smoking and even smoking e-cigarettes, for those of you who are doing that. But there is intervention. The gold standard is ventilation treatment. So you might have seen this during the COVID period on various news items. It's a quite invasive way where you've got a ventilator, you've got compressions happening on the chest, sometimes it's prone style, and essentially it's a very unpleasant way to treat it. But it does treat it for a short period of time. So this is what the lung looks like after a short treatment of ventilation. We've got a nice clear alveoli, we've got our gas exchange restored, the breathlessness is restored. We can see we've got these sort of restored some of our lung function, essentially got rid of the water. The problem is that's not a pleasant treatment, and it's often associated with post-intervention issues. So fibrosis, inflammation, even more edema. So we end up in this vicious cycle, and these patients get very, very sick with it, both mentally and physically. So these are the patients we were looking at, and to sort of get a feel for this, I have been, as part of the clinical trial I'm working on at the moment, I've been interviewing patients, and so we interview them when they come in for their ventilation treatments, and a lot of their stories really give you a feel for it. So imagine right now I asked you to stand up, walk to the very back of the lecture theatre, walk to the front, and then sit down again. So you'd be a bit cross with me, you wouldn't really enjoy that, but you'd hopefully be able to do it. But for these patients, even walking from this side to this side of the room would not be possible for them. They would have to sit down. They would be breathless. They would be fatigued. And their stories sort of mimic that. So one patient said, I have to sit down 5 to 6 times when I'm walking up the stairs to my bedroom. And he hastened to add it was one flight of stairs and not a big flight of stairs. Another said, I couldn't even stand for a few minutes to dance with my daughter at her wedding. And another said, I used to run 10K and then play b-ball, basketball. Now I can't even stand still for more than a couple of minutes without needing to sit. So we have this group of patients who are really suffering, and our treatment for them is not brilliant. So we set about in the US with my postdoc, we were looking at pharmacological intervention. How can we treat these patients and offer them some alternate? So to do that, we sort of looked more closely at the lung. If you think about a nice healthy lung, we've got a nice open airway. This is our alveoli. This is what we want to see. Nice shape, and more importantly, we have a nice intact blood vessel. But in these patients, we tend to see catastrophe. We see large amounts of inflammation. We see fluid accumulating in the alveoli. So this area right here, where they should be exchanging oxygen and carbon dioxide, is obliterated. This is what gives them the breathlessness and makes it so hard for them to move and to even stand. And specifically, we're interested in this region here. My interest is how the blood vessels can stay intact to avoid this breakdown that we see here. How do we keep a nice, stable blood vessel? So we looked at what causes the blood vessel to be stable, and there's lots of junctions that hold the cells together. The one that is most important, the one that we literally cannot survive without, is adherence junctions. And this is like a zipper. So if you imagine kind of putting your hands together, I've got one cell here and one cell here. And this zipper is an anchor. It holds the cells together. It means that we get a nice barrier that is formed for our blood vessels. It keeps everything intact. And if you think of without that barrier, then fluid can physically move across. So we looked at all around how you can control that. And we found lots of different proteins. We found lots of different proteins that when you activate them, when they move around the cell, that they can cause a tight barrier and give us that sort of nice junction. And we found some that actually they do the opposite, they cause more leak, and we understand the mechanism around that. And that was brilliant. It was great because we got papers, it was great because we understood more about these important proteins. However, for those of you who are biomeds in the field, you might recognize that a lot of these proteins are not specific to the lung. In fact, they're important for every single cell type. So we can't go around manipulating any of these proteins in these patients, or we're going to start affecting all sorts of other parts of the body. So they weren't a real therapeutic option. They were interesting and important, but they didn't take us further. So armed with this experience, armed with my papers, armed with my funding, armed with some incredible researchers I got to work with in the US, I thought about, right, what's next? And if any of you have been to the East Coast and had to dig your car out, after a while it gets frustrating and boring. It's not pleasant. Our winters here are a bliss in comparison. So my fellowship ended and I decided to move back to the UK to avoid any of this. At a similar time, my sister and brother-in-law introduced my nephew and niece into the world, and I decided to, hopefully in the field, I decided to take responsibility for their science education. I felt it was imperative. Just after that, I met my husband and convinced him to move to Cambridge. And I joined ARU. So what do I do when you join ARU? I was an ECR. I was still finishing up some papers from my PhD, which some of you may kind of have empathy with. This was a few years on, but that's what happens, right? Papers sometimes take a while to come out. And I was trying to figure out, what do I do here? I've got this experience with the lung blood vessels, and I want to carry on that. I really enjoyed that. I also have these other areas, the kidney and the signalling around that was really interesting. And I'm a new ECR trying to find my feet. So what does any discerning scientist do at that point? Well, they get themselves to the canteen. They get some artificial sweeteners, which they know stimulate the taste receptor. They get a cell model of the human lung. In this case, these are blood vessels that are taken from humans, and the blood vessel cells are isolated. So we had this nice model in the lab. And we start looking for the taste receptor. We start looking at effects. So we found the sweet taste receptor in the lung and in the blood vessels, which was a really interesting sort of new step. But the big kind of step forward for us was a year later when we got this data. So what we're looking at here is we're looking at our cells, and our cells from the blood vessels, we can mimic that in the lab. So they form a nice tight barrier. They all sit nicely together. And we can put something called LPS on it, so a bacterial protein. And we can measure how leaky that blood vessel becomes, or how leaky those cells become. And so where we see this big drop with our model of lung injury, we see a lot of leak, a lot of fluid is moving across. When we came in with our activator for that receptor, we saw complete blocking of that leak. So instead of seeing sort of the lung injury side of things, we saw a nice, tight, intact barrier. And this was really exciting, and this was sort of something new, and I still had a lot of the connections and collaborations in the US. So we spoke, and we got an animal model to investigate this more. So the animal model is quite similar. It uses a type of bacteria, Pseudomonas, and we see this leak in the lungs. And this is very much what we would expect to see in patients as well. Hopefully you can see the difference between the two. Instead of having a nice white intact set of lungs, we've got a lot of blood accumulating. We have fluid. It's quite heavy because it's so full of fluid. And we can study this model, and we can have a look. And that's exactly what we did. We just measured how heavy the lungs are, how much wet weight there was, how much edema there was. And so what we saw was that with our model, our bacteria, we saw leak, we saw edema, water on the lungs. And when we came in with our sweetener, we saw, similar to our cell model, we saw a decrease. And over time, we've looked at different sort of mechanisms. How is this happening? Why is this happening? So we know from the literature, from our studies, from other studies, all the different molecules, all the different proteins that are what causes our vessels to become leaky. So what we did was we had a look at these, and in every single case, when we come in with our activator, we are blocking those signals. So we're doing something here in the blood vessel that is cancelling out the injury that we know happens in lung injury. So again, really interesting, but what does it actually mean? So we've taken it forward to patients who have ARDS. So with collaborators in the US, we have a clinical trial. And if you think about patients that I described who can barely walk from sort of one side of the room to another, they typically have to come into hospital all the time. They have to come in for ventilation therapy, they have to come in for checks, they have to come in for various different types of steroid treatment to try and help them. And so with this trial, we are adding IV of our sweetener to the trial to understand it, the effects it's having. And we have the advantage that sweeteners are everywhere. They're FDA approved, they're EFSA approved. We're not talking about a brand new drug that has been developed. This is something that is in the market that we can get for 20p. So we studied this, and we have taken a lot of different measures, but the one I want to talk to you about is the oxygen saturation. So you'll probably have seen this before. This is the peg that will sit on your finger, and it will measure how much oxygen is in your blood. So this is a kind of crude way for us to look at ARDS, but it's also a very typical way. So if you think about the normal is sort of 500 millimeters of mercury. That's what we'd expect to see. ARDS patients are all the way down the bottom here. So we're talking about very little oxygen in their blood and very high mortality, unfortunately. And with our treatment at 6 months, we are seeing reversal of this. So if you think about going from an ARDS model, we're going from severe to moderate. So what that means, it means the risk of mortality is dropping by nearly half because they have more oxygen that they're getting in their bloods. And more excitingly, at 12 months, we are moving to a mild or asymptomatic form of ARDS. So they still have ARDS, but in all the clinical classifications that we have, things like mortality, ventilator-free days, biomarkers, we are seeing an improvement. We're not back to normal yet. We haven't fully analysed all the data to understand what this means. But if you think we're taking very sick, very vulnerable patients to a place where they are doing better, that has to be a positive thing. And as part of this, I get to interview these patients after treatment. And so if you think about the person I told you about who said he had to, 5 to 6 times he had to sit down before he could walk up a flight of stairs. In the most recent interview, when he was part of the 12-month treatment group, he just come back from a long walk on the beach with his wife and his dog. He said that he was tired, so he did need to take a little nap, but he felt good. And that's got to be an improvement. I mean, to take somebody who was struggling to move in terms of quality of life, fantastic. So I know you're sitting there thinking, hang on a second, Javi, you're saying you can treat this horrible disease with a can of Coke or Pepsi or other soft drinks are available. Certainly I've thought that. Or like me, you were thinking, hang on, go to Pepsi or Coke or whoever it is and ask them for some money because this is groundbreaking. Unfortunately, like all things, it's not that simple. There are a lot of negative effects of artificial sweeteners. So at the same time that we were doing this and we were uncovering this and we were setting up our clinical trial, I had a very talented PhD student join me, and she was a microbiologist. I was trying to convince her to be a physiologist, but she was having none of it. And we looked at artificial sweeteners. There were some studies suggesting that the gut microbiota, so the bacteria in our gut, were susceptible to artificial sweeteners, and they might be having a negative effect. So we jumped on that, and we had a look in more detail. And Aparna spent her PhD looking at this. So we looked at the cells in our gut to see what effects the sweeteners were having on the cells. We looked at the bacteria, some models of different gut bacteria. We even looked at patients from a community in Bangladesh who have diabetes to look at different effects on their gut, or different effects that sweeteners were having on their gut. And we routinely found this negative impact. We routinely found that the bacteria became more pathogenic. We routinely found that our own cells would either die or just become more stressed from exposure to artificial sweeteners. And actually, this research was included in some news outlets, but also in some guidance that has come out from WHO and from the UK government around using sweeteners. And so we know it's not a straightforward story. We know that we can't rely on artificial sweeteners, certainly from an oral consumption point of view, to be any kind of good. And this is work that Aparna and I are carrying on, and actually I'm getting to do with Carrie Walker as well. So we're trying to understand this more, to try and figure out how artificial sweeteners are having this negative effect on the gut, how they are having this negative effect on the bacteria, and also whether there's things that we can do to reverse that. Because I don't know if you in the supermarket have looked at a packet lately, but almost everything has got artificial sweeteners in it, even things you wouldn't expect, like a packet of crisps. So we are exposed to these, and we need to understand the effect they're having on us. So we're now in this weird balance where we know there are negative effects to the sweeteners, but we know we have these really promising effects with patients from the ARDS side. So we're taking the clinical study further to understand that better, to understand the limitations of where we can take that, but we're also looking at other options. And one of the things we're looking at is bitter receptors. So if we think about sweet receptors, we've got them here in the middle. There's only 2, maybe 3 at most. But with bitter receptors, we've got loads that we can look at. And we're starting to understand, trying to understand the different bitter receptors we have in our lungs and whether they could be a better target for us. This was work that was done by Susanna and carried on by Lewis. And we're trying to figure out if these will offer an alternate. As is the case with science, it may not be. If I can talk to you in 5 years' time, I hope to be able to have some data to show how promising these could be. So alongside my research story, I've had the opportunity at ARU to be a lot of different things. So I joined as a senior lecturer. I've got to lead modules. I've got to lead a course. I became associate professor. I've got to work with a fantastic School of Life Science team. And most recently, I'm just finishing my tenure with Claudia as co-deputy head. And I'm afraid this is where it's going to have to get a little bit mushy, because Claudia has been my touchstone. I absolutely, and I don't mean to be hyperbolic, I wouldn't be standing here if it wasn't for her, because I wouldn't have even thought I could apply to be professor. Her inaugural talk is in February, so please come along. It won't be as good as mine, obviously, but I'm sure it will be fantastic. So I have to thank Claudia. And we met when we were waiting at a bus stop in Cambridge, so, there was a lot of time for us to get to know each other, but 10 years of friendship and co-leading the research in the school has been a fantastic experience. Anyone who is interested in job share, I would say, do it. It has been a wonderful experience. And the other 2 people that I have to say exactly, us academics do love to wear our graduation gowns. Nick, you have been a fantastic support and friend. And Chris, your face has been in every single professor talk that I have been to. So there is a whole family of professors, a whole generation of us who stand on your shoulders, and your mug is up in every single one of them. So I think that's got to say something good. Thank you for being a fantastic mentor and colleague and friend. So, as I say, apart from learning that academics love to wear graduation gowns, and we do every year, you'll see us there brightly coloured, I wanted to just kind of touch on a couple of things that I've learned on the journey, other than, you know, all the great research that I've got to do. I have been told that I have a finger in a lot of pies, which I think means that I'm a jack of all trades. I think it's a nice way of saying that. Whilst construction is not, you know, the right avenue for me, I tried it. As you can see, I have no idea what I'm doing with whatever that machine is. I have had the opportunity to work with a lot of fantastic people on a lot of great projects, and I do love to do that. So I wanted to give you some examples of that, and I wanted to kind of say to any young researchers in the field, take the opportunities, because for every new project you are definitely learning something new. With the sports scientists, Justin, Ash, Sanjoy, George, Joe, thank you for coming, and with Lata from the forensic science side, we have done some fantastic work in looking at supplements and looking at the effect they have on cardiovascular disease, on, sorry, not on cardiovascular disease, on cardiovascular markers of exercise. And so these guys are doing fantastic work. This has been a side avenue that has actually turned into one of my favourite projects. I've also got to do some fantastic work with Mohammed, Mary and Aisha, based down in Hems, looking at cancer. So taking a little bit back to where I started, but in a different way, looking at different cancer therapeutics. With them, I am learning about chemistry in a way that I have not done in a long time. So that is brilliant. Shahina, who has taught me that the blood vessels in the eyes are almost as interesting as the blood vessels in the lungs. And we're looking at ways to use that information to try and think about patients with diabetic retinopathy and age-related macular degeneration. Nick, who your mug has to show up again, and Emily, who were doing some fantastic work on platelets. So from the cardiovascular side, I will always be a blood vessel biologist, but things inside the blood vessel like platelets can be interesting. Yulia, Danny, inspired by Danny, and with Evie, who's a fantastic project student, we've looked at e-cigarette-induced lung injury. And so this is sort of the other side of lung injury, which is the kind of causes, and trying to understand better why that happens and how that happens. And that's obviously a very modern kind of type of lung injury compared to some of the other ways we've looked at it. Yulia is a fantastic fellow who is teaching me about AI as we look at cardiovascular disease predictors and ways of treating that with, through an ARC fellowship. And then Richard Jones and Maggie, and we're doing work on 3Rs. So how can we take animals out of research? How can we find other ways to reduce the amount of animals that we use in research? And Maggie's doing fantastic work over at Cambridge University on that. So these are seemingly very disparate areas of research, but for all of them I've been able to kind of lend my expertise, and I've had the opportunity to learn so much. So I am happy to say that I am a jack of all trades. I will take that title because it has given me the chance to work with fantastic people. I did just want to say there isn't enough time, but I've been lucky enough to be a member of, supported by, or on a range of different committees, funded by lots of different sort of academic institutes, research institutes, publications, and this is part of probably my jack of all trades, and it has been a wonderful thing. I wanted to end with a kind of sort of summary, I guess, and I wanted to say that you can be what you can see. So a couple of years ago, we were sitting in this room, and someone who shall remain nameless gave a fantastic professorial talk. And it really resonated with, I know, a lot of us. And it was all about sort of role modelling in science and looking around you and you know, who is there. And I think the lesson that I would say is I have been incredibly lucky to be surrounded by strong, intelligent, brilliant women, also men, but I think the point around you can be what you can see. I can be what I can see, both personally and professionally. I've been lucky to be mentored, inspired by everyone, all the way through from my grandma, mum and aunts, down to my 9-year-old niece, who is arguably the strongest female that I know. So on that note, I wanted to say thank you very much for listening, and I'm very much honoured to be here.
Thank you so much. A really inspiring, a really wonderful, inspiring lecture. Very thought provocative. I'm sure we've got questions, but thank you so much for your really profoundly collegiate approach, both in what you do, but also in recognising so many colleagues. Thank you very much from me. So, Havovi's kindly agreed to take some questions. Tallest man in the room this afternoon.
So, couple of statements and maybe a question in there. So I'm so wonderfully proud of you. It's fantastic that you quite rightly achieved your professorship and well deserved. So that's that bit. Get that bit out of the way. And I've forgotten what I was going to say next.
That's fine.
No, here we go. So you showed this fantastic data how treating odds with the sweeteners in patients is improving their oxygenation. But then there's this other side, this is bacterial side of things. What does that mean? Does that mean that these commensal bacteria are then becoming infective and that are, is it life-threatening to patients? And do you see that in all patients that are treated with this, or is there differences? Do all patients respond in exactly the same way that you've showed that patients, or is there a full spectrum of what's going on there?
No,
and do you get resistance?
It's a really good question, yeah. So we haven't looked at gut bacteria in our patients because these are patients who are very sick with lung injury. But the work that Aparna did looking at different model gut bacteria shows that with every type of sweetener we look at and with the different types of model bacteria, that we're always seeing that the bacteria can stick to and invade our own gut cells. And so that's linked to things like sepsis, even down to multiple organ failure. What was interesting is with the patients in a small group of diabetic patients we looked at in Bangladesh, they were complaining of symptoms that were very similar to things like Crohn's or IBS, which again links to that sort of... So we've seen that, but what's interesting is the field is seeing that as well. And actually there is quite a few studies now suggesting that artificial sweetener consumption on a regular basis can dysregulate our gut bacteria and actually make us more likely to get diseases like diabetes. So I think we're very clear on that side of it, on the consumption side of it. But what we're not clear on is, do the benefits for these patients outweigh that? Would IV treatment actually mean we wouldn't get that? Exactly. Yeah. Or do we say actually it's something we'd be willing to live with because of the benefits it could give? Haven't seen it, haven't looked for it. I'm going to now look at the data and check, but as yet we haven't seen it. We've gone as far as 12 months. Our next trial is longer, yeah, very early days and very small numbers.
Thanks, Havovi. Appreciate it.
Thanks, Havovi. Great talk. A really basic question. The magnet or the amount of sweetness that would produce a good effect and the amount of sweetness that would produce a bad effect in the gut, for example, how much is that really? Is it like 1 can of Coke, 10 cans of Coke?
Yep, so the treatment we gave to patients was equivalent to 5 cans of Coke. That's per treatment, so that's not per day. That's every time they came in, which for some patients was weekly, for other patients it was monthly. So that doesn't sound, that sounds like quite a lot. But actually, if you think about what people consume, maybe unknowingly throughout the day of artificial sweeteners, it's not that ridiculous, it's not that high up. So when we see damage to the gut from artificial sweetener consumption, it's not dissimilar to what we're giving to the patients. It's actually quite a similar concentration.
I shouldn't ask, but I need to ask before I go back. Surely there's a wide range of artificial sweeteners. Are all these benefits and risks equally the same for all of them?
From the artificial side, yes, so saccharin, sucralose, they all affect our receptor, so they all have that effect. We're starting to look at things like stevia, monk fruits, the sort of natural sweeteners in quote, but we think they work through the same receptor, so they're likely to have the same, both negative or positive, but we haven't found that yet.
Thank you. Sorry.
Thanks, Howie, for a great talk and for showing us how it's done. My question, I think you sort of hinted at this, but then didn't develop it further, is by saying that, at least orally, I think is what you said, are there any mechanisms for trying to get these treatments into the lungs, but then not into the gut?
Yeah, so that's what we do with the patients. We give it IV because we have the unfortunate luxury that they are coming in for IV treatment. So we're already in that scenario. With our next trial, as we move forward to patients who are more in the moderate to mild group, so we're seeing if we can push them even to a better scenario. With those, we're going to have to find an alternative because they're not necessarily being IV'd, so we're not necessarily going to be able to do that. And so we will have to find a way other than orally. And that's why it's on my mind.
Okay, so non-biologist in the room, though I did do some a long time ago. Are these artificial sweeteners absorbed by the gut? So they do move into the blood?
In small amounts.
Okay, but a smaller dose than that's consumed.
Yes.
Possibly naive question. Why are there these receptors in the lung and the kidney? What are they doing in the normal, non-treatment, non-RDS body?
This question keeps me up at night. You don't know. This is the million-dollar question, so it's not naive at all. My feeling is, from the lung, there is some work from the bitter receptors that suggests that it can be in response to bacterial proteins. So, there's always one, Susanna. So we think that for the bitter side, that these receptors are there because we have bacteria and bacterial proteins could be stimulating it. But from the sweet side, we have no idea. And what is more confounding, what really keeps me up at night, is that you need large amounts of glucose to stimulate these receptors. So larger than we would ever have in our blood. But you only need tiny amounts of artificial sweetener because they are acutely sweet. So why they're there, I mean, they've been found now in the bone, the bladder, the brain even, some people have shown, the heart. And the heart was really cool because the paper had sweetheart, which I thought was quite, love a pun. So we don't know, but my feeling is it might be something to do with that. These are large proteins. This is difficult for the blood vessel to make. They're not going to make it unless they need it. But why it's there, we don't know yet. I'm willing to take suggestions.
Hi, thank you for your wonderful talk. I just wondered, has anybody thought or tried using nebulizers to administer the treatment for ARDS?
It's a really good question. The problem with the nebulizers is that it hits our epithelial first, our alveoli, before it would get across. Would it get across, and would it get across to our blood vessels? Because we don't think this is affecting the alveolar. We think this is specifically affecting the blood vessel. So that's why we think the IV is the best route. Looking at different treatments for ARDS, nebulizers only help with sort of indirectly to help patients breathe. They don't help with delivery of drug.
Thank you. There's a question here. There is a mic coming. Would you be kind enough?
Oh.
Sorry. We'll come back to you. I'm just giving this chat the best work out I can.
Here.
Hi there. These sweetener receptors that you find in so many organs of the body, how do they compare, or are they similar to the sweet receptors that you find in the more conventional taste buds in the tongue? Is it the same molecular mechanism, or is it a different process?
Yeah, it's a really good question. So in terms of the protein themselves or conformation, we think they're the same. But in terms of the signaling, they seem to be working through slightly different ways. So in some cells, they seem to be working through calcium, in others through cyclic AMP. So each cell type seems to have a different response. So for example, with the calcium, that's more to do with muscle contraction, so in the bladder contraction or in the heart, whereas what we think is more cyclic AMP route that's happening in the lung. So it seems like in different tissue they're responding differently. That might just be that in different cells there's different machinery is there. It might be specific to the receptors. We don't know.
Thank you. And I think we should probably make this the last question.
All right, thank you for your speech. Please, I want to know, what is the effect of this sweetener in the cell cycle? Because I am trying to really understand cell cycle, sweetener, reduction, oxygen carrier capacity. How do we correlate that?
Yeah, so we, in the lung blood vessel cells, we don't see a change. In the intestinal epithelial cells, we saw that the sweetener causes the cells to die. We didn't look at cell cycle, so we don't know specifically if it's through that mechanism, but we see a lot of oxidative stress in the intestine from the sweeteners, so we think it's through a sort of stress mechanism, but we haven't looked at cell cycle.
Well, thank you so much. Thank you for all your excellent questions. I'm sure you have more. There's a do I say wine? There's a liquid and food.
No sweets.
Opportunity afterwards to further thank and congratulate Havovi. I am now going to pass to Laurie and ask Laurie to formally thank Havovi for her wonderful talk on behalf of the faculty. So Laurie, please.
Havovi, thank you so much. I mean, just a truly wonderful talk, really, really interesting talk. You covered so much in about 45 minutes. It was truly, truly impressive. So, I mean, I just thought there were a number of things that really sort of struck me. One was that non-linear academic journey. I mean, it's not the first time we've heard that story, but it is such a powerful story, and yours, of course, is unique, and, it really, really impressive the way in which you describe sort of your travel from actually being able to predict at the age of 10, I think it was, that you're going to become a professor of biomedical science, which is truly unique. I've not come across that before. But the fact that actually the rest of that journey through to becoming a professor, it wasn't all just a continuous linear journey, but it did have, bumps and challenges in the road. I just thought that was really good and very honest to present that. So thank you for that. And I thought also the really sort of the powerful, patient-focused research, curiosity-driven research as well that underpinned the major theme of your research. I just thought that was absolutely fantastic. And, hearing patient testimony, which, I wouldn't necessarily expect to hear from, a biomedical scientist studying physiology, it's just fantastic. And it just joins everything up together in a way which was really compelling, but, really, really just shows just how important the research you're doing truly is. And then I think the final thing I just want to take this away from this was the jack of all trades comment at the end. That really did resonate, because I think I would describe it as a connector of people, of ideas, of disciplines, which is an absolutely essential linchpin of really, really great research and great researchers. So I think it's not just a sign of a great researcher, I think it's also a sign of a really great leader as well, which I think you undoubtedly are. So I think hopefully all of us will take a little bit of that away and hopefully try and improve ourselves as jack of all trades as well. So on behalf of everyone here, thank you so much. I've got just a little gift for you as a sort of a token of our appreciation. But thank you so much. Thank you. So, as the Vice-Chancellor said, we have, I think, liquid and things next door now. So, if you'd like to join us for a drink afterwards to talk further with Havovi and friends and colleagues, please do that. Thank you very much for supporting these events and coming this evening, particularly if you're a first-time visitor. We have, as Havovi queued up earlier on, we've got a number more of these events coming up over the next few months, so do keep an eye out for those. We do also, next week, our unique Inaugural University, we have the Chelmsford Science Festival kicking off for a week next week. So if you are minded to do so and able to travel up to Chelmsford, then please do Google Chelmsford Science Festival. It's all around AI this year, power and perils of AI, so do take a look online, and if you can attend one of the events, you'd be most welcome to do so. But apart from that, thank you once again for attending